In this class, we'll discuss about insulin secretion. This insulin secretion is done by beta islet cells, and these beta islet cells are present in pancreas. And these beta islet cell will act as a glucose sensors, and this glucose sensing activity is linked to secretory machine of beta islet cells. After post-meal, blood glucose levels will be increased. Whenever blood glucose levels are greater than 70 milligrams per deciliter, then insulin secretion will happen. Now, this is a beta islet cell. Whenever blood glucose level reaches greater than 70 milligrams per deciliter, now this blood glucose will enter into beta islet cell through a specialized transporters. These are facilitated diffusion transporters. These transporters are called as glucose transporters, which help in facilitated diffusion of glucose. And there are different GLUT transporters, but GLUT2 will help in transport of glucose from blood to beta islet cell. One more GLUT is GLUT1. These two will help in transport of blood glucose from blood to beta islet cell. In the beta islet cell, this glucose need to be converted into glucose 6 phosphate with the help of glucokinase enzyme. This glucose can be converted into glucose 6-phosphate by two enzymes, either glucokinase or hexokinase. But beta islet cell will have more glucokinase activity. Why glucokinase? Because this glucokinase has high Km. That means it has low affinity for glucose. Because of low affinity, there is no feedback inhibition, and glucokinase activity is directly proportional to glucose concentration. That's why glucokinase enzyme is used in conversion of glucose to glucose 6-phosphate. Next, it enters into glycolysis and it will form pyruvate. This pyruvate will be converted into acetyl CoA. This acetyl CoA will enter into PCA cycle and ATP is formed. And as glucose entry increases, more of ATP is formed. Next, we have to discuss about potassium channel. This is called as a potassium channel, and this potassium channel is made up of two subunits. One subunit is called as Kir6.2, which is an actual potassium pore, which causes flow of potassium from inside to outside the cell. And there is SUR1 subunit, which is a sulfonylurea receptor, and this is a regulatory subunit. Now, what is the normal function of this potassium channel? It causes extending of potassium from inside the cell to outside the cell. Now, whenever glucose levels are increased, this glucose entry also increases and causes more of acetyl-CoA, more of ATP formation. This causes inhibition of this potassium channel. Due to inhibition, now potassium levels will be increased inside the cell. This potassium increase will causes depolarization the membrane, and this depolarization will activate L-type calcium channels. These are L-type, L-type voltage gated calcium channels. These calcium channels are activated by depolarization. Now, calcium will enter from outside the cell to inside the cell. This insulin will be stored inside the granules. Now, this calcium will act on these granules and they will cause exocytosis. Exocytosis. This exocytosis will release insulin. Along with the insulin, C-peptide will be released. Now, this is the main pathway of insulin release. There are amplifying pathways also where other mechanisms will help in insulin release. We have separate receptors called as incretin receptors. These are called as incretin receptors. These incretin receptors are acted by GAT hormones. GAT hormones. These GAT hormones are called as incretin hormones. They are nothing but GLP-1 and GIP. They directly act on incretin receptors, and these incretin receptors will cause exocytosis of insulin granules and causes release of insulin. And there is one more function like HNF. These are called as hepatocyte nuclear factor. This HNF also causes insulin release. Okay, insulin release is done with the help of HNF. What is HNF? This is hepatocyte nuclear factor, causes insulin gene stimulation and insulin release, and they causes glucokinase gene stimulation and causes increased glucokinase, and they upregulate GLUT2 transporters. Upregulation of GLUT2 transporters. This is how HNF will be causing insulin release. And this HNF mutations will cause separate disease called as maturity onset diabetes mellitus in young. And we have, this is how insulin will be released, and this insulin released is in two phases. Insulin release will be in two phases. First phase, second phase. In first phase, it is between 0 to 10 minutes, and this is from 10 minutes to 60 minutes. And this is from pre-formed granules, and this is from stored granules and new insulin synthesis. These preformed granules are near to membrane, and these stored granules are away from the membrane and new insulin synthesis. So this is rapid and sharp and short span, and this is sustained insulin release. And what is the specialty here in type 2 diabetes mellitus? First phase is lost early in the disease. This is how insulin secretion will happen after post meal. Thank you.